In the last section, we talked about different ways of studying the brain, and a huge amount of information has been accumulated by these and a few other methods. The human brain is a complex three-dimensional object. Viewed from outside, you can see a major separation between the left and the right cortical hemispheres. You can also see a structure called the cerebellum at the lower back, and the cerebellum also has two hemispheres. There's a brainstem that emerges from the bottom, which connects down to the spinal cord. The cortex is a thin sheet of neurons that covers the outside of the two hemispheres. And there's also what's called a cerebellar cortex, that's a sheet of different types of neurons wrapped around the outside of the cerebellum. So that's what you can see from outside. But as I said, there's a lot of very complex physical structure that's hidden inside. One of the problems talking about the anatomy of the brain is describing where different structures are located relative to each other. So there's various terms used to indicate different types of relative position. Anterior means more towards the front, while posterior means towards the back. Dorsal means more on top, while ventral means underneath. Lateral means more out towards the side, while medial means more in towards the center. And finally, it's sometimes useful to refer to the two ends of a structure as rostral and caudal. Rostral is towards the part of the structure that's more like a head, and caudal is more towards the tail-like end. There are 8 major anatomical structures that are going to be central to our discussion of higher cognition. The first is the cortex. The thin sheet of neurons that makes up the cortex is about 2 millimeters thick, and it's separated into 4 or 5 layers containing different sizes of pyramidal neurons. There are different areas on the sheet, and there are slight differences in the organization of the layers in the different areas. Then, the neurons are also organized into columns perpendicular to the layers. And there's more connectivity between neurons within a column than between neurons in different columns. The sheet that makes up the cortex is wrapped around the outside of a mass of axons. Most of these axons are coated with a layer of a white chemical called myelin, and as a result the axon mass is called white matter. Now sometimes we also speak of grey matter by referring to the mass of neuron bodies. However, grey matter only looks grey after death. As I pointed out earlier, in a living brain it looks red because of blood circulation. In the hippocampus, there's also a sheet of neurons called the CA fields. The neurons in this sheet are the same as in the cortex, but there's only one layer. The hippocampus also has a V-shaped structure called the dentate gyrus that has a different type of neuron, in fact a couple of different types. A nucleus is a clump of neurons that's separated from other neurons by regions that contain mostly axons and very few neurons. Often, a major nucleus is made up of lots of smaller or subnuclei that are separated by smaller neuron-sparse regions. The thalamus is a nucleus, and within the thalamus there are a number of smaller nuclei, and almost all these subnuclei have very similar neurons that are quite different from cortical neurons. The basal forebrain is also a group of similar nuclei with more different types of neurons than the thalamus. The basal ganglia is a group of nuclei including striatum, globus pallidus, substantia nigra, subthalamic nucleus. Each of these nuclei have different types of neurons, but because there's a striking pattern of connectivity between these nuclei, the basal ganglia is regarded as one structure. The amygdala is again a group of nuclei, but some of those nuclei have cortex-like neurons and may even show a very simple form of layering. Others have neurons like those in a part of the basal ganglia, the striatum in fact. The hypothalamus is a cluster of a large number of very disparate subnuclei with considerable differences between the neurons in the different subnuclei. And the cerebellum is a very complex structure with an outer sheet that has 2 layers. Each layer has different types of neurons, and the sheet is even more folded than the cortex. And like the cortex, it surrounds a body of white matter. And also like the cortical hemispheres, there are groups of nuclei in the middle. And then there's a couple of nuclei down in the brainstem called the pontine nucleus and the inferior olive that are functionally also pretty much a part of the cerebellum. There are some additional structures that we'll also touch upon, including the raphe nuclei and the locus coeruleus, which are small groups of neurons in the brainstem that target extensively throughout the brain. So what I want to do next is to give you a feel for how all these structures fit together in an actual human brain. This is an MRI image of a sagittal slice of a human brain. This slice is vertical, goes from front to back, and is somewhat offset from the center. Many of the major structures can be seen in this image, and I'll indicate the approximate locations of the ones you can't actually see. The most obvious is the cortex. The sheet of neurons that makes up the cortex has a total area of about 3 sheets of regular letter paper. The sheet is crumpled up to fit inside the skull. So imagine this crumpled sheet wrapped around the outside of 2 large masses of axons that mostly provide connections from one part of the cortex to another. And as I mentioned, these axon masses are called white matter because the axons are coated with myelin, which is white. And the myelin coat makes signals travel much faster along the axon. When you crumple up a flat sheet, you get ridges and hollows or valleys. In the cortex, a ridge bulging out is called a gyrus, and a valley going in is called a sulcus. And about 2/3 of the area of the cortex is buried in the hollows or the sulci. The folds differ somewhat between different individuals, but there are some major folds that appear relatively often or always. I've indicated 2 gyri and the sulcus between them that tend to be present in every human brain. The cortex sheet wraps around all of the 2 hemispheres, including much of the region between the hemispheres where they face each other. Where the cortex goes up between the 2 hemispheres at the back, it thins down and curls around in a spiral to form a structure called the hippocampus. The hippocampus isn't really visible in this section, but I've indicated its rough location, and you can see it more clearly in this horizontal section on the left and the coronal section on the right. And of course, there's a left and a right hippocampus, so I've circled one of them in each of these 2 sections. The thalamus is very centrally located in the brain. Just as for the cortical hemispheres and most other brain structures, there's the left and the right thalamus, and it has the same subnuclei in both the left and the right sides. Information from the eyes and the ears only reaches the brain via the thalamus, with very few exceptions. Information about body position and movement also reaches the brain via the thalamus. So the vast majority of sensory information only gets access to the cortex via the thalamus. And the major exception is the sense of smell, where receptors in the nose are directly connected into the cortex. But in addition, the thalamus also manages the internal information flows between different areas of the cortex. The basal ganglia is a group of nuclei mostly located in front of and around the sides of the thalamus. Unlike the thalamus, these nuclei contain different types of neurons, and in one case, a nucleus is located some distance from the others down in the brainstem. But because of the striking pattern of connectivity between all these nuclei, as we'll get into later, the basal ganglia is regarded as a single structure. It plays a big role in initiating body movements, but it's also important for cognition. This is the rough location of another major structure called the basal forebrain. Part of the basal forebrain, called the septum nuclei, communicates heavily with the hippocampus. Other parts of the basal forebrain communicate very widely with the cortex. And this is the rough location of the amygdala. It's adjacent to both the cortex and one of the nuclei of the basal ganglia, and the amygdala is heavily involved in emotions. The hypothalamus nucleus is located near the bottom of the cortical hemispheres. It's connected to the pituitary gland even lower down, which acts on the body by releasing hormones into the blood. Different hypothalamic subnuclei influence behaviors and activities like eating, drinking, body temperature regulation, sleep, emotional behavior, and sexual activity. One subnucleus is the mammillary bodies, and these mammillary bodies seem to play an important role in memory. The cerebellum is this heavily convoluted structure located below the back of the cortical hemispheres. It has an outer layer of neurons called the cerebellar cortex, and the cerebellar cortex surrounds a volume that's mostly axon white matter and is buried inside of that white matter is a group of nuclei. So in a general sense, there's a resemblance to the cortical hemispheres, but the neuron types and the connectivity are very different. And the cerebellum plays a role in the detailed coordination of movement, and it's become clear that it also plays a role in speech and in cognition. A number of structures like the pons are located lower down between the cortical hemispheres and the spinal cord in a region called the brainstem. All the direct communication from the cortex to the cerebellum, and there's a lot of it, goes via a part of the pons called the pontine nuclei. And another subregion of the pons, called the locus coeruleus, is one of those small groups of neurons I mentioned earlier that send axons all over the cortex. The spinal cord is the bundle of axon fibers that carries communications between the brain and the body. Within the brain, there are also some major paths that carry lots of axons. The corpus callosum is a nerve bundle that connects the left and right cortical hemispheres, and it has about 200 million axon fibers. And by way of contrast, the nerve bundle connecting the eyes to the brain contains about 1 million fibers. The nerve bundle carrying information from the cortex down to the cerebellum has about 20 million fibers. In other words, it's smaller than the corpus callosum, but still a lot larger than the communication between the eyes and the brain. The fornix is another major nerve bundle. It connects the hippocampus to the basal forebrain and to the hypothalamus. The fornix has about 1 million axon fibers and is very important to memory. There are also a number of liquid-filled ventricles in the brain that don't appear to have a strong information processing role. They provide structural support and perhaps chemical distribution. The reason I mention them is that they're useful landmarks for locating other structures. So, having gone over these major structures, in the next section we'll get into more detail about how they're positioned relative to each other.